Hi and Assalamualaikum. I hope everyone's watching very well today. So our group focused on the analysis on food safety and biomonitoring of Polymedosa erosa and Polymedosa expansa. So the result in this analysis are based on its dry weight. Hi, I'm Norazi Mokhtar. I'm going to give a little information about this analysis. The information of heavy metal levels in Malaysian aquatic environment is very limited and always mainly focused on the peninsular Malaysian area. So in Malaysian waters, these mud clams act as benthic filter feeders, and this indicates the potential of biomonitor of heavy metals. This can be said because the animals ingest metal-enriched particles directly. And next, the objectives of this analysis.
Hi guys, so to begin, we're going to talk about the body parameter that we had specifically analyzed was the dry tissue weight, two species of Galoina, and their heavy metal concentrations. So, we have Polymesoda erosa and Polymesoda expansa, and we'll dive into the erosa analysis first. So 18 individuals of Polymesoda erosa were sampled, and they were clustered into 9 clusters with 200 grams of size intervals. So I'll zoom in. Next, 6 different metals, as you can see, zinc, cadmium, iron, copper, plumbum, and nickel. They were also analyzed in the dry tissue weight, and the mean and standard deviation were graphed. As shown in this figure, erosa dry tissues had a high amount of iron embedded in the dry tissue, followed by zinc. And other heavy metals were below detection limit, so they were deemed insignificant. Next, we're going to dive into Polymesoda expansa. So, in contrast, so it's the same as erosa, as the heavy metal concentration were highest in iron and also in zinc.
Next, we're going to discuss the food safety guideline for clams consumption for Galoina species. Here is some of the example of food safety guidelines on heavy metal for different countries and organizations. In this part, I will focus on zinc concentration for Malaysian Food Regulation 1985. And we can also see that almost all the example here does not list the guideline for iron. From the graph, we can see that the concentration of zinc in Polymesoda expansa exceed the guidelines for the cluster 1 with the size interval between 0 until 0.199 gram according to the Malaysian Food Regulation 1985. By using SPSS, we are able to analyze the correlation between the dry tissue weight and the heavy metal accumulation for these two species. From the graphs, there is no significant correlation between the dry tissue weight and the heavy metal accumulation because the two graphs are negatively distributed. For the correlation data obtained from SPSS, we can see that the Pearson correlation for Polymesoda erosa is negative 0.431 and Polymesoda expansa is negative 0.797. Both of these indicate that there is no significant correlation between the dry tissue weight and the heavy metal accumulation under the 0.05 significant figure. A large sample size, at least 50, is needed for both species and an even distribution of individuals in each interval cluster size group. This will help to increase and strengthen the correlation between the dry tissue weight of Galoina species and the heavy metal accumulation.
Okay, next we move to determination the biota sediment accumulation factor. First, for sample Polymesoda expansa, we using this formula where biota and sediment sample were taken from Penggalat Papar. Okay, this table shows the BSAF sample for P expansa. And graph heavy metal again BSAF according to the cluster size. Next, for P erosa. Okay, this table is BSAF for P erosa according to cluster size. We get this after we calculate by using BSAF formula just now I mentioned. Okay, and this next. Any significant according to cluster size group. First, for P erosa, okay, before that, to know the significant difference, we using ANOVA to find P value. As you can see here, P value at rows are less than 0.05. Thus, there are significant differences to cluster size group for P erosa. While for P expansa, as you can see here, the p-value at row are more than 0.05. Thus, there are no significant differences to cluster size group for P expansa. Okay, question 6. Could polymer sodium SP be a good biomonitoring tool? If yes, for which selected heavy metal and why? Yes. Polymer soda SP is a good biomonitoring tool for zinc and copper. This is because zinc and copper has had high mean concentration and had the highest value of BSF compared to other heavy metals. From the graph, bioaccumulation process occurs many times in small cluster size. It's shown in cluster 1 and 2 at zinc. There is higher value of BSAF. In bioaccumulation, if cluster size is small, means more heavy metals can be taken. Therefore, the smaller the cluster size, the higher the rate of heavy metal taken. So, this estimation currently shows that the consumption area of P erosa or P expansa could cause no toxicological risk to the consumer, and it is useful to suggest this species as a good biomonitor. However, further studies are still needed.
So based on Nor Atika's explanation before, we can answer this question. Does this biomonitoring tool applicable in other locations? And the answer is yes, because there are many past research and experiments have been done. Can also act as a water filter. So there are some proofs here we can see based on the past studies done. The comparison of heavy metal accumulation in P erosa between the two locations. And next is the heavy metal accumulation in P expansa between two locations. So those two graphs can tell us that the water in Johor has high content of heavy metals compared to Papar Sabah. Now I'm gonna reveal the mechanism and the depuration process. As you can see on this figure, inside the shell. As you can see here that's the mouth. And let's straight away go on to the process. The accumulation and depuration of heavy metals occur in this species are by filter feeding. So during the feeding, these clams usually bury themselves in the sediment, as you can see, to protect themselves from predator and water current lah. When the tides come in, Polymosoda species will extend out the inhalation siphon and suck the fresh water containing planktons and other waterborne nutrient contaminants. Then the accumulation process takes place especially in highly polluted water. So the water enters via the inhalation siphon, then it passes through the gill plates that is filled with mucus, trapping all the heavy metals inhaled. and flushed out filtered water via the inhalation siphon. Depuration only occurs when polymer soda are in clean water. Clean water washed out the trapped waterborne particles in the gills. So that's when the tank depuration done in laboratories is for food safety and food security management. In this tank, it is to remove the pathogens, contaminants, and impurities through Clem's very own natural depuration process. They are kept in clean waters instead of polluted water, so nothing will be filtered through it. Instead, it will be washed out again. This is to prevent illnesses to the consumer.
Next, to reiterate what Liu had said, our country has set our own limits in the Malaysian Food Regulation 1985, but how can we implement this in our local communities and also in communities in other parts of the world, especially ones that live near the estuaries? Well, first, mud clam agriculture projects can be introduced by the Village Development Committee, and these projects boost socio-economic status. At the same time, they ensure that the consumption of mud clams are monitored to avoid risk in human health, especially in these specific areas. So next, in these areas also, women play a large role in risk management. For instance, in the Solomon Island, as you can see in the picture, women in the community are in charge of gathering the clams, thus they have a certain protocol and actively manage the protected areas there. Moreover, the fisheries department in each state is also responsible to educate and train the villagers to harvest and select mud clams based on the size and the maturity, which this particular practice will aid sustainability and reduce health risks that go with this improper consumption of the Galoina SP. Next, in general, the management in agriculture harvesting will bring structure and sustainability for the future of mud clam harvesting and to successfully manage the food security in important areas. In conclusion, according to the data that we had analyzed, a larger sample size is needed for the Polymosoda erosa and Polymosoda expansa as to obtain better and more significant data. And these two species we have found by the BSAF ratios that we had calculated is that they are good biomonitors for zinc and copper, especially in Papar Sabah, as you can see here. And these mud clams in the area are safe to consume as they are below the limit stated by the Malaysian regulations. And although since no guidelines were set for iron, it is better to consume with caution in order to not get iron-based diseases that will affect your immune system and affect your health. So that's all of the objectives in this study were successfully achieved. So these are our list of references for further reading. And thank you. Let's move on to the Q&A session.